Click the bell icon and subscribe to our channel so that you will not miss any classes. Hello, welcome to class. In this class, let us discuss the next requirement or the tool in recombinant DNA technology, that is the host. That means whenever we have created a recombinant DNA, here it is a vector, to this vector we have attached the gene of our interest and now it is called recombinant DNA. And this recombinant DNA should be expressed, that means it has to multiply and it has to produce the product. This gene product should be produced, and for that production it requires some host. And in biotechnology there are various hosts which have been used. For example, the host like animal cells can be used as a host. Plant cells can be used as a host. Even bacterial cell, not even. Commonly bacterial cells are used as a host. And fungal cells, especially yeast, have been used as a host. The thing is, whichever the host we are using, say for example, it is of any kind. This is a cell, host cell, it is of any kind. The requirement is it has to uptake this recombinant DNA. It should have the capacity to uptake this. That means this recombinant DNA should enter or should integrate into the genome of the cell and it should get expressed. So for that capacity, they will call it as competent. If the cell is capable of uptaking the recombinant DNA and integrating it into its genome and expressing, and that cell is called competent host. It is called competent host. All the cells are not competent. Some cells will have natural competence. On that course, there are two types of competence we can say. Natural competence and induced competence. I'll talk about first one, the natural competence. First one, natural competence. Natural competence is placed back to 1900s when Griffith conducted an experiment on mice using Streptococcus pneumonia, the bacteria. You might have remembered it from molecular basis of inheritance, where Streptococcus pneumonia is a bacteria. It is present in two forms. One is S form, or we can say it as S strain and R strain. So when he was conducting some experiment, S strain is pathogenic and it was causing disease in mice and responsible for death of mice, and it is because of the presence of polysaccharide capsule. And R strain is not pathogenic. Whenever it is injected into mice, it was not causing any disease. That means there are some genes responsible for production of capsule in S strain and those genes are absent in R strain. So when he conducted a series of experiments, trial and error he done, he came to know at some point that R strains of bacteria have been converted into S strain. That means they must have received the genes from S strain. That is, he put them into a solution. I am not going to the details of this experiment. So when he combined these two, actually he killed it. He killed the S strain and it combined with the R strain, live R strain. At that time what happened? R strain was converted into S strain. And for that he given a conclusion. Some of the materials were present in heat kill S strain. Upon heating they have not inactivated. They have been like that and they have entered into R strain and converted the R strain into S strain. Which means there are some materials and those materials we know them as now DNA or the gene. These genes were entered naturally into R strain and converted. Not only entered, they integrated into the genome of R strain and converted that R strain into S strain. And for this procedure or for this process, he called it as transformation. Transformation. For this simple process, we can give the definition as it is the process of transfer of DNA into the bacterial cell. Okay, that is called transformation. This is that means our cells are naturally competent enough to uptake this DNA. So it is the natural competence. Okay, many of the bacterial cells have this natural competence. Naturally they can uptake the DNA. But this is found to be somewhat inaccurate or time consuming process. Biotechnologists may not depend on this natural competence for every kind of host, which means there are range of hosts available. So they may not be competent enough to uptake the DNA as like Streptococcus pneumonia did. So for that they have to induce any of the host cell we are using, we have to induce the competence. So why? You may ask the question why. So I will explain it now under the heading induced competence. That is creation of a competent host. Okay. So induced competence, or we can study it under the heading creation of competent host. Why there is a necessity of creating a competent host is DNA molecules are hydrophilic. They are attracted towards water and moreover they are negatively charged. And all of the host cells, whatever we are using, they have lipid bilayer in their cell membrane. We have studied the fluid mosaic model of plasma membrane. We know that there is presence of lipid bilayer. Okay, the host cells have lipid bilayer. So because of this lipid bilayer, negatively charged hydrophilic molecules of DNA cannot enter into the cell easily by passing through this lipid bilayer. It will repels. Since the lipid bilayer is hydrophobic, it will repels the DNA. Easily it will not allow the DNA to enter into the cell. So for that reason, we have to make this cell as competent cell. And irrespective of which type we are using, either we may use plant, animal or bacterial or fungal. Okay. So for this induction or induction of the competence in the cells, there are various methods available. So the common method used is treatment of host with divalent cations like calcium or magnesium. So magnesium ions can be added to the growing plates of bacteria before their usage, that is half an hour or 1 hour before the usage, so that it will create the pore. So this is not a commonly used method. Usage of calcium ions for inducing the competence is a commonly used method, and that method is popularly known as heat shock method or calcium chloride treatment method. Calcium chloride treatment is the commonly used method for inducing the competence or preparation of competent cell. So here what they will do is, they will make a solution, a defined concentration of calcium chloride will be added, and they will make a solution of calcium chloride. So this is a solution of calcium chloride. Okay, so which will be carrying the calcium ions. In a solution, calcium ions are released, dilate. And here then add the host cells. The host cells will be added to the solution. So these are the host. So when the calcium ions binds to the membrane of the host, and after adding the calcium ion, this chamber should be incubated on ice. So it should be incubated on ice. That is at very low temperature. So at this temperature, calcium ions binds to minute pores present on cell membrane. So cell membrane already have the minute pores. Okay, they are very little. So for that little pores, the calcium ions will bind. When the calcium ions bind, the pore size will increases. Okay, when the calcium ions binds to minute pores present on the plasma membrane, the pores will dilate. Pores will dilate. Dilate means they increase in their porosity. That is, the pore size will increases. Okay. So after this, to this solution, they will add the second step. So this is the first step I read here. So host will be added with calcium chloride ions and in the form of solution, and it is incubated on ice, during which the calcium ions will bind to the minute pores present on the cell, and it will dilate the pores. Pore size has get increased. Now, the next, what happened? After dilation of the pores, I will write it as, this is second step, incubation on ice. And the third step is, to this solution containing calcium chloride and the host cells. And the host cells, I told now, they have dilated pores. Pore size is increased. Then to this, the recombinant DNA will be added. So recombinant DNA will be added. So think what will happen now. The recombinant DNA is added, then the recombinant DNA will enter into host through dilated pores. Through the dilated pores, easily the recombinant DNA will enter into the host cell. Okay, so the entering of recombinant DNA into host is only not important, but also it should integrate it into the genome of the host. Okay, so here this cell, the host cell will also have the genome, right? Into that genome, the recombinant DNA should enter. So for that, what they will do in the next step, this solution will be given with the heat shock. So that is, it is placed under ice, right? So after previously it will be under ice, and suddenly the temperature will be, or the solution will be brought to the temperature of 42 degrees Celsius. So this is called heat shock. Heat shock means so not too much high temperature. It is the temperature of 42 degrees Celsius. So when this is given, later again the solution will be bring back to ice. It should be incubated on ice again. So this treatment, that is cold, hot, cold treatment, will allows the integration of recombinant DNA into the host cell successfully. So this completes the preparation of competent host. So have you remembered now how we can create the competent host by heat shock method or calcium chloride treatment? Solution is prepared of calcium chloride and host cells will be added to it and incubated on ice, during which calcium ions will bind to the minute pores present on the host cell and it will dilate. After dilation of the pores, recombinant DNA will be added to the solution. Then it should be recombinant DNA will enter into the host cell through the dilated pores. Then for integration of recombinant DNA into the host genome, heat shock is given at 42 degrees Celsius and again it is bring back to the ice. Okay, so this is the method of creating a induced competent or preparation of competent host for taking the recombinant DNA and expressing it. So further things we shall discuss in the next class.